This week, the U.S. Patent and Trademark Office granted a CRISPR-Cas9 genome editing patent to research teams for Jennifer Doudna and Emmanuel Carpentier. What does this new patent cover?
So this is a very interesting patent that covers a way of using CRISPR, in particularly Cas9 CRISPR, in a setting outside of a bacterial cell. So it's important to think about how Cas9 assembles inside a bacterial cell. Inside that cell, a long transcript is made, and a second piece of RNA pairs to that. The Cas9 binds to that pairing, almost like beads on a string, and then the individual RNAs are trimmed so that the Cas9 is left with two RNAs inside. The way everybody uses Cas9, however, is they provide the two finished pieces of RNA, or the two pieces of RNA joined together, but it's trimmed down from how it would appear in nature. And what the seminal work in 2012 from Doudna and Charpentier revealed in their labs was the minimum requirements to get that to assemble and function in a setting where you're providing the components exogenously, not how it's normally made inside a bacterial cell. And it turns out that is, of course, how everybody who uses CRISPR-Cas9 now does it. They don't express it in a bacterial cell, they provide the elemental components and the components assemble. And the rules for designing the RNAs to get them to work in that setting was what was discovered. It is not how it occurs in nature, and so that is the basis of the patent that was granted this week, the patent that was issued this week.
Do you think there's any potential for using this patent as a basis for the arguments in that they're trying to, I guess, overturn the Broad Institute's patent?
So what is notable about this patent is it's distinct from the IP that's currently under litigation. So it's been judged patentably distinct by the PTO, particularly in light of the existing litigation. So this is considered distinct subject matter compared to the patents that are under litigation. So it stands apart.